Okay, so we're thinking about the Calvin cycle, and that first enzyme in the cycle is Rubisco. And here they're representing it by that blue. So this is the enzyme, that's the active site. And you can see RUBP is about to drop into the active site. And then there's two options because Rubisco never evolved the ability to have a high binding affinity just for carbon dioxide. For some reason, it's still, after all these millions of years, it still is okay with taking in carbon dioxide or oxygen into its active site. So that's what you're seeing here. And if it's carbon dioxide, that's great because then you get two phosphoglyceric acid molecules, which can feed further into the Calvin cycle. And that is awesome because then the plant is able to make its sugars. Here, if oxygen comes in, then we get one PGA, which is good, but then that phosphoglycolate is only two carbons. It can't fit into the Calvin cycle. Instead, it has to do the PCO cycle, which is what we're looking at here. And so we looked at this last time, how you have RUBP right here, and then you can see oxygen coming in. So those two are reacting. And you end up with one phosphoglycerate, which can feed right into the Calvin cycle. So that's good. And then this two carbon phosphoglycolate. That has to go through all of this. Now, actually, this cycle has to run twice. You see the number 2 in front of all these molecules here. So basically, what they say is the PCO cycle has to run twice in order to build one more phosphoglycerate. So by the end of the PCO cycle, you do get a payoff of another phosphoglycerate, another PGA. But it's a lot of effort. You can see 3 different organelles involved, lots of different molecules. You have CO2 in the mitochondria actually given off instead of getting fixed into sugars. So that's not good. So in general, these plant physiologists say this is not a good thing to have running constantly or even modestly. And when they do controlled experiments where they make a plant do this, it really is not good for the plant. Low yield, low biomass slows the growth rate. It's more stressed. So it kind of makes you wonder, why, why would the plant be doing this? And there's a few things to be aware of. If you think about plant cells, which are mostly water, so in the cytoplasm, you have mostly water. In the chloroplast, the watery portion is the stroma. So if you have CO2 and oxygen dissolved in the cytoplasm of plant cells, and it's about room temperature, under those conditions, you have more CO2 compared to O2. It's a 3-to-1 ratio. So that 3-to-1 means you have more carbon dioxide than oxygen, which is good. That's good. So if you have a situation, there's two ways to think about this. If your oxygen levels are low or if your carbon dioxide levels are high, either way is fine to think about it that way, then you're going to get carboxylation going on, which means basically what we mean by that is Rubisco will take in CO2 into its active site and react that with RUBP. However, if you have the reverse where oxygen levels build up or if CO2 levels crash, then you're going to get oxygenation, which means oxygen's going to come into Rubisco's active site. And then you'll have to run the PCO cycle. Another thing to be aware of is that when the temperatures increase, think about, around here it'd be like July when we start to get into the 90s. If you have increased temperatures, what happens is in the chloroplast, in the stroma, where you have dissolved oxygen and dissolved CO2, once you have that plant experiencing higher temperatures, in general, the solubility of gases decrease at higher temps. That's kind of a universal thing, no matter what you're talking about. But oxygen will be less affected. So basically, what that means is as you get a hotter and hotter day, carbon dioxide starts to bubble out of solution and be lost to the plant, whereas oxygen sticks around longer, dissolved. And so basically, you're creating a situation where Rubisco is starting to see more and more oxygen and less carbon dioxide. So thinking about plants who experience warm temps on a pretty regular basis, this is a big risk. If they don't have a way to deal with this, they're going to be running a PCO cycle a lot. And so this is something to think about. So basically, as the environment temperatures go up, you're going to have oxygen interacting with Rubisco a lot more. Another thing to be aware of is under drought conditions or high salinity, some crops, some fields where we grow crops, for example, are pretty saline now because they've been over fertilized, and fertilizers are essentially salts. And so these fields have become too 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 much of the leftover fertilizer, high salinity. And then under drought conditions as well, we're definitely have periods of drought across the nation and around the world. That also is favoring Rubisco seeing too much oxygen. And again, kicking in the PCO cycle. So think about areas in the world where there's routine droughts, and then with a lot of crop plants, we have the salinity issue. So all of this is creating situations with, when you think about these last two bullet points, basically it's tipping the scale so that Rubisco is seeing more oxygen than carbon dioxide. Now you have to appreciate also that Rubisco evolved way back even before plants were on Earth. There was algae in the lakes and cyanobacteria and, in the oceans and lakes, but no higher plants yet. And when Rubisco evolved, when the cyanobacteria were around, there wasn't oxygen initially in the atmosphere. There's a lot of CO2, but there wasn't any oxygen millions and millions and millions of years ago. So if Rubisco evolved under those conditions, there was no selective pressure to worry about oxygen, if you want to think of it that way. So they often point to this, that, that's one reason why it can accept either or. But you would think over millions of years, it might have tightened up a little bit, but it just never did.
Was gonna say, even though it came early, wouldn't you think that Rubisco, if it causes this much like negative effect,
Yeah.
why wouldn't it evolve to get around it?
Exactly. And
Which poses, does it benefit in some way?
and that's the question a lot of physiologists are asking right now. If Rubisco never tightened up to ignore oxygen, maybe there is a reason for it. And so one that they point to is this one, that maybe when you look at the PCO cycle, maybe some of those intermediates that you see are actually used in other pathways that are important. And so they're chasing that down. They're doing research on this. There's a lot of research on Rubisco. And Rubisco isn't the only enzyme that's kind of sloppy like this that can take more than one thing into its active site. But it's a fairly short list of enzymes that do this, at least that we know about anyways. And so when you look at the intermediates here, I mean, there's a lot going on here. Maybe some of these are important in some, you can imagine these shooting off into another pathway. So it could be. And they are definitely, it's interesting debate going on in some of the published papers on this, where they're saying maybe this is not always as bad as we think. But having said that, now if you start thinking about plants that might live their life because of where they are geographically, they might live their lives in consistently warm temperatures or droughty areas. And I mean, if you've traveled to any of these places, I'm sure you've seen that there's still plants around. And so what happened is instead of tightening up Rubisco, plants evolved a different strategy. to try to stop the PCO cycle from running, especially running frequently. And so we're going to talk about those. And once you understand this concept, it helps you to appreciate when you're traveling around and you see these plants, it's like you'll understand why they're so successful in certain habitats and perhaps not in others. So we're going to talk about two different cycles or pathways. One is called the C4 cycle, and one is called the CAM pathway. And you can see the pictures here are very different geographical habitats. We have more of a tropical or subtropical is the idea when you think about the C4, and then really harsh desert conditions where you have long drought periods. Okay, so let's think about the C4. They call it C4 because the first stable molecule in the pathway in the cycle is a 4-carbon. So that's why it's called a C4 carbon cycle. So we're thinking about plants that are in consistently warm conditions. And so think around the equator and just north and south of the equator. You have the tropical, subtropical regions. And when you have plants that are native to those habitats, so they evolved in those habitats, they have a very different leaf anatomy compared to a plant like from around here that's native to this area. And so we'll look at that. You're going to see that these C4 leaves have their mesophyll cells inside, and then there's their bundle sheath cells. They're very different looking compared to what we see in a typical leaf drawing. I'll show you all this. And we're going to find out that the C4 cycle does a spatial segregation of where things happen. And we'll look at that too visually. So we say it's a spatial segregation of different reactions. RxN is reaction. Now, C4 plants also have a C3 cycle. The C3 cycle was the first cycle to evolve in how to make sugars. So all plants have a C3 cycle. It's just that some plants tacked on these extra cycles on kind of and linked into the C3 with it. When you look at these plants that are native to tropical, subtropical, they can handle drought and hot temps and water stress better than just a straight C3 plant. Excuse me. And so even though they're in tropical areas, there are periods of drought. It's not as extreme as a desert, but they do have some fluctuation. And when they've done studies on them experimentally, they say, yeah, about 2 to 3 times photosynthetic capability. They're better at doing photosynthesis. They're just more efficient. They're faster at it compared to just a straight C3 plant. And basically, excuse me, when you look at the C4 cycle and you kind of, follow it all the way through and say, what's going on here? What's the benefit of doing a C4 cycle? This is it. It concentrates CO2 around Rubisco. Okay, so it creates a situation where it says, I'm going to make sure that Rubisco is seeing lots and lots of lovely CO2 and hardly any oxygen. And this cycle accomplishes that really, really well. All right, so that's often how they refer to these. They call them CO2 concentrating cycles, really. Okay, so let's look at some pictures. So here you can see the leaf in the top. And that is a C3 leaf. I'm looking at it going, what? Wait, no. And then I forgot. It's a C3 leaf. So that's typically how they draw leaves. And that's good for a C3 plant. So there's a lot of C3 plants out there, and you can see their leaf anatomy. Okay, now let's look at the C4. So there's the C4 on the right, and you can see a little different anatomy. You can see that the spongy mesophyll cells are more abundant. You don't see really a palisade. These are palisade cells. You don't really see that showing up in the picture on the right. And the other thing the artist is trying to show is that the bundle sheath, which is this ring of cells, the bundle sheath cells that surround the vein, are really beefed up. They're plump, bigger, more chloroplasts. Okay. The other thing that should have been drawn a little bit better is that typically with C4 plants, you see the spongy mesophyll cells kind of hugging up better to the bundle sheath. The artist was not quite accurate on that. I think I have some better pictures here in a minute. Okay, so basically what they did now is say, okay, let's look at a bundle sheath cell and a mesophyll cell and blow that up. So what you're seeing on the right is the way in which they spatially separate these reactions in two different cell types. So you can see here that the mesophyll cell kicks it off. The stomata open at sunrise, CO2 comes in, and you can see that it reacts with PEP, not RUBP. Okay, so the C4 cycle, you have CO2 and PEP reacting, and it's a totally different enzyme initially. It's not Rubisco. It's this enzyme, PEP carboxylase. So that it's named that because it takes CO2 and carboxylates, adds the CO2 to the PEP molecule. PEP carboxylase doesn't care about oxygen. Oxygen will not fit into its active site. It ignores oxygen. So this is great. So even under hot conditions, when CO2 initially comes into the plant, it's immediately going to get hooked up with PEP. And then the immediate product is oxaloacetate. Okay, I would like you to know what the immediate product is. That's why it's named a 4-carbon, the C4 cycle. It's because oxaloacetate is 4 carbons. Okay, then once oxaloacetate, you can abbreviate that down to OAA. Once that's formed, it just does a simple rearrangement to malate. Okay. And then you can see that malate physically moves into the bundle sheath cell. So that's why I wish the artist had put, because you have to have spongy mesophyll cells kind of hugging up to the bundle sheath cell for this to, whoops, easily happen. Okay. Then when malate comes in, there's two things going on here. CO2 gets cleaved off of malate. So CO2, and this is what we refer to as when we said it's a CO2 concentrating capacity. This is what we're referring to. The malate has that CO2 coming off of it and it gets fed into the Calvin cycle right where Rubisco is. So basically, you have to imagine Rubisco in this bundle sheath, okay, right where the Calvin cycle is happening. Rubisco's sitting there and malate comes in and says, here's some CO2. And it just keeps doing that. The cycle keeps running all day long and it just bathes or surrounds Rubisco with a bunch of CO2, thanks to malate moving from the mesophyll to the bundle sheath. Then once that happens, once CO2 comes off of malate, the other product is pyruvate, which is good because now this is a cycle, the C4 cycle, so we have to come back to having enough PEP. So then pyruvate shifts back to the mesophyll in order to make PEP. You do see ATP is needed for that last step. So this is not a free reaction. This cycle, it costs some energy, but the benefits outweigh the costs. Okay. Questions on the C4? I like to think of malate as in science, we often talk about vectors, how vectors are used to move something from point A to point B. If you think about like mosquitoes as vectors, right, for malaria. And so they carry the malaria pathogen. And so they often say, mosquitoes are a vector for malaria. I think of malate as a vector for CO2 because really malate has a CO2 molecule as part of its makeup. And so when malate is moving, it's moving from point A to point B. It's acting as a vector in order to deliver, instead of giving somebody malaria, it's delivering CO2 to Rubisco. And if this keeps happening all day long, Rubisco just keeps seeing a ton of CO2. The other really good thing is that that first enzyme that interacts with CO2 does not care about oxygen. It has the fancy way to say that is it has a really high binding affinity for CO2. Yes.
Is is going through the meso, like the, you know, I don't know how to say this. Is the bundle sheet cell like, is the only way it can get gases through, is it through the mesophyll cell? Like is, because we said that the first part ignores oxygen, so is it only carbon dioxide that's going into the bundle sheet cell?
Yes, that's a really good point. So the other thing that I haven't told you yet is that here, let's see. Okay, it's not really on here at all. It's not obvious on here. So when you look at this, the way the artist drew this, you see that the Calvin cycle is only showing up in the bundle sheath, okay? So what's going on in C4 plants is that the mesophyll cell basically is doing the light reactions of photosynthesis. What's that one product that we really don't care about when we think about what the plant's trying to accomplish with the light reactions? Oxygen. We care about it. So you have to appreciate that in the mesophyll cell, there is oxygen building up here. But the way the artist drew this, in the mesophyll cell, you don't see the Calvin cycle happening, right? So what's happening is basically most of the Calvin cycle really is happening in the bundle sheath cell only. There's a little caveat to that we get into in plant phys, but I'm not going to get into it here. So the key reactions of the Calvin cycle are happening only in the bundle sheath. So that means Rubisco is hidden away from oxygen that's being built up in the mesophyll, potentially being built up. All right, so they kind of hide Rubisco away from where the light reactions are going. And then on top of that, malate delivers CO2 into the bundle sheath. So by the time, as this is going on all day long, the bundle sheath has a situation where there's very low oxygen because there's no light reactions going on in the bundle sheath. And then malate's delivering a bunch of CO2. So you've tipped the scale where you have lots of CO2 in the bundle sheath and very little oxygen. Okay. Yes.
Do you think that in the mesophyll cell, do you think it evolved to have that to get around the Rubisco?
Mhm.
To like,
Yeah.
to stop it from grabbing all of the oxygen?
Mhm.
Like
Yeah.
block it?
I mean, it's really amazing how they had to really shift, like devote, okay, we're only going to do the Calvin cycle and the bundle sheath, and so they had to really kind of switch gears through the evolutionary process. And, they, through the process of that and lots of adaptations to what was going on, they were able to, finesse it to the point where we see it today of Rubisco's tucked away into the bundle sheath where no light reactions are really happening. And then you have PEP carboxylase, which is really, really good at only paying attention to carbon dioxide. So you have a lot of things that have tipped into favor of Rubisco seeing lots of carbon dioxide, very little oxygen. So really quite an amazing process. This one, this is just, again, what we've been talking about. If you want to know what PEP is, there it is, phosphoenolpyruvate. Okay. And that PEP enzyme PEP carboxylase enzyme is very efficient, which means it has a very high binding affinity for just carbon dioxide. It doesn't care about O2. Whoops. And so you really don't, you really don't see the PCO cycle happening very much at all in C4 plants. This figure also has a little bit more detail of some of the molecules. Well, it doesn't, oh, there it is, oxalo acetic acid, that's why it's abbreviated OAA. Malic acid, and you can see again, malate serves as the vector. Okay. This is what a C4 leaf looks like in reality. I believe this is corn or maize. And so you can see how the spongy mesophyll are hugged up a little bit better to the bundle sheath cells. You can see how big those bundle sheath cells are that surround the vein. Really plump, full of chloroplasts. The spongy mesophyll surround them nicely. Here's another image. C3s are, C4 is on the top. Okay. And this artist did a little bit better job where they're showing how the spongy mesophyll and the bundle sheath. So you see there's nice, the blue is the bundle sheath. You see that they're hugged up nicely. So they can pass those molecules back and forth. So if you think about plants that are native to tropical regions, corn, maize is definitely a big one, considering how much corn we grow across the United States. Sugarcane, I have it in the notes, sugarcane, some other grasses, Poaceae family is grasses. And so sorghum is another one that's gaining a lot of attention in the food industry as well. So that sorghum is down here. That's sugarcane. So some pretty big crop plants. When they look at it, and they have been investigating what plants do this, it's across a lot of different families of plants. So it's not dedicated to just the grass family, for example. It's 19 different families, and it can be monocots and dicots. A lot of species in the grass family, the Poaceae, used to be called Gramineae, but now it's been renamed to Poaceae. Asteraceae, we'll talk about them later in the semester. Adding it all up, it's about 3% of all known species can do this. Unfortunately, some weeds are really good at it too. Weeds that show up in your garden or your lawn. And they think it happened about 70 different times in terms of the plants tweaking it and going through natural selection. So there's been a lot of effort at this over the millions of years. Okay. This is a quote, C4 plants, 40% greater ability to convert sunshine into biomass compared to just straight C3 plants. So this quote, you can understand why a lot of people who grow plants for a living, crop production in particular, they're like, huh, wouldn't it be nice if all of our plants could do this, where they're that much more efficient? So think about rice, for example, how much that's being grown globally and how much that feeds, how many people that can feed. And so this project has been going on now for quite a while. The C4 rice project, it hasn't reached completion, but they are trying to get rice plants to become C4 plants. That means they have to genetically, engineer and tweak it. That's quite an undertaking. My hat goes off to them because that's quite an undertaking to convert a straight C3 plant and say, let's make it do a C4 cycle. So we're working on it. Okay, I'm going to stop there. On Monday, we're going to cover the CAM cycle, which is, thankfully, you're going to see some of the same molecules involved. We're going to talk about PEP carboxylase again and OAA and malate and all of that. All right, so it's a lot of the same chemicals. It's just a different way of, instead of a spatial segregation, CAM plants do a temporal or a time segregation of when they decide they're going to do certain things throughout the 24-hour period. So we'll get into that on Monday, but let's stop there so we can talk about the exam a little bit. Did you see that I sent out an announcement that there's a little